So I actually don't know if it's gonna, yay, okay. It was just because I had the little pen thingy clicky. Okay, so chapter 3, we're going to try and do quizzes over, so maybe we won't even have a major exam. Like, you guys are like 3 and 4, how is that going to be presented? I despised the McGraw-Hill thing, so I'm glad other people thought it was educational and beneficial. And they're hopefully group texting you important facts that we covered. Okay, so chapter 3, and there is an important topic. So we colored the cell, but what are these? You guys remember these? Yay! So these are phospholipids, and when we put them together, we want to remember that there was like the fatty part that was the main barrier. And then because we had water inside a cell and water outside of a cell, then the water-loving heads would just automatically face outward. And so anytime that we're working with the cell, so just imagine that we have a large cell here, it's really wrapped in a phospholipid bilayer. And we just want to take that construct and remake them like, oh, there's a lysosome, and it's a phospholipid bilayer. And we had a rough ER that was studied with ribosomes, and it's a phospholipid bilayer. Like, we're not building it out of new stuff. If there's a barrier, it's a phospholipid bilayer. So we just want to remember to reconstruct that in our minds over and over and over again. So our nucleus was in here, and it's actually a double phospholipid bilayer. So mitochondria is the same way. Like here it's a more succinct double barrier, but this inside here is a phospholipid bilayer, and out here is a phospholipid bilayer. So it is a double again, except it leaves 2 different spaces. So like an inside the matrix and then. So those things are really important just to understand functionally what is crossing that membrane. And that if we have a vesicle and we fill it with like proteins and it comes out here, if it's made out of phospholipids and the membrane's made out of phospholipids, it can just merge. Does that make sense? Because it's all just these loose phospholipids. So like can get along with like just fine, and it just kind of merges and dumps its stuff outside the cell. Or this phospholipid can kind of pucker in and take in the liquids. So all these kinds of nifty things that the cell does is based off of this phospholipid structure. So what other words can we describe that phospholipid bilayer with? They, them, the like greater world beyond. Yay! So like anyone, teachers, other classes, other instructors, the textbook, what would they call it? So cell membrane sometimes is called a plasma membrane. Any other words that you've heard? I'm like, I have, but fluid mosaic model is what the whole thing is called. So this phospholipid bilayer being a structure, they use that term fluid mosaic because it's really just these little component parts sort of floating in water. So it's not a solid physical structure, more like a raft that just completely surrounds the cell and makes a big bubble. So that is just the name of the model, like what this, I guess, theory or however you would explain it. It's a modeling of that system. Okay, so the fat is the impenetrable layer. What is it penetrable by? Repulsing water, so nothing water-loving can get through, but what can get through without any kind of permission or channel. Okay, so like if lipids, the impenetrable layer, the things that can get through are like the cholesterols and sex hormones and lipid-based chemicals. Like when you go fuel up your car, the reason that you cannot wash that gas smell out if you get gas on your hands is because it has absorbed into your cell. So definitely want to protect yourself from things like that. But the thing that can penetrate is the lipids. So the only barrier is to water-loving things. And what we do is we just put these little channels. So it's usually drawn something like that, that we make it possible to go into and out of the cell via a channel. So some are regulated and some are just open. So I'll draw a really tiny one here and we'll call it an aqua. So we actually have pores or little fenestrations or windows through, however you want to explain it to yourself, but they're called aquaporins. So they're little protein channels that are tiny enough. They don't really regulate the flow of water, but so just open pores that are eligible for water to go into and out of the cell. Okay, so do you guys know of organelles that do not have, that were labeled on? Yay. So ribosomes are actually little proteins, right? So they're just proteins. They do not have a phospholipid bilayer. And there was one more, and it is named as an organelle. It's not like a standalone. Yay! Okay, so the nucleolus is a section that's just darkened inside of the nucleus, and it is ribosomal RNA. So it is the protein's area or the gene area to make ribosomes. Like it's the script, and it just looks kind of like a darkened region. So nucleolus and ribosomes are the two that I can think of. Microfilaments and microtubules I also think are just proteins. Okay.
So
extracellular fluid? No. Okay.
See, I can imagine
yeah.
other things.
Well, I mean, water can actually go to and from pretty freely with these aquaporins, but we don't let our nutrients just go willy-nilly wherever they want. So we pump in nutrients and pump out products of the cell. So our nucleus is busy making those RNAs and like messenger RNAs and things like that, and that's going to go directly out here. We're building proteins inside. We'll release them in a vesicle, but we will keep a lot of that stuff inside of our cell and use it in the cell versus dumping it outside of the cell. So the concentration inside of a cell has a lot more formed elements and electrolytes and enzymes and things like that than the fluid outside. It's more water-like. The channels, yes. So we have active transport and diffusion.
Yeah, like the aquaporins are just
open. So water can go to and from pretty freely. It's just small enough. Yeah, it's like water is much smaller than sugar or something like that. Couldn't
so
just.
It's just simply the size.
It's just too. Yeah, it's just like gonna block the hole like a finger in a dam or something like that. Yeah, does that kind of make sense? On a molecular level, I just am just guessing on a magic school bus kind of level there that's what that's like. I don't really know. But I know that it needs a much larger protein channel to move potassiums, sodiums, chloride ions inside and out of the cell. So it might be charge-based because a water molecule is not extensively charged. It just has a slight polarity. Whereas like a chloride ion would be way negative and a potassium would be way positive. So maybe the charge is kind of regulating it from going in and out. Yeah. So don't know exactly the answer. I do know that it will not be flowing anything inside and out of this cell other than the water, we'll just imagine, can go fairly freely. But even it needs a channel. Other good questions? So we can kind of understand the phospholipid bilayer thing? Yeah? Okay. So I'm going to